All right, thank you so much.
All right, thank you so much, Caitlin. So, as she said, I'm Midhula Gireesh, and I'm the assistant professor and extension specialist, and my responsibilities are mainly turf and ornamentals. So we can talk about red-headed flea beetle today, which is an emerging problem here in Tennessee. So I'll talk about the beetle, the overall the introduction, the identification, overall gentle biology, and then we'll transition to Karla. So just a heads up. Okay. All right, so what is red-headed flea beetle? So the scientific name is Cisterna frontalis. So it is a beetle, so the order Coleoptera and the family Chrysomelidae. It's a major pest problem in condator ornamental plant nurseries in the central and eastern US. So they have like a wide host range, and they're also sometimes referred to as the cranberry flea beetle and also the blueberry flea beetle, based on what host. That is a testimony, like they have like a wide host range too. So why red-headed flea beetle matters? Number one, as I was saying before, the increased reports in Tennessee and the southeastern US. Like as of now, we are hearing a lot of problems in Tennessee about this pest, because that was not a problem in the past, because we don't have a lot of container production. But in the nearly, like we are seeing like a lot of reports from red-headed flea beetles damage and also the beetles as such. So again, as I said before, it's a problematic pest in container grown nursery production system. How? They are extensive like foliage feeders. So the damage is mostly the feeding that they cause to the foliage, and it also reduces the aesthetic quality. It's mostly like the damage is aesthetic, but overall it reduces the sellability of the plant and the marketability of ornamental plants. So that is why it matters a lot and why we need to know more about this pest. Okay, a little bit history and distribution. So they are not native to North America, and they are widely distributed across the eastern and the central US, as I talked before. And their early records primarily from wild and weedy broadleaf host. And historically, they were considered as a minor or sporadic feeder. And in the past 10 to 20 years, they've become like a major economic concern. And at least they are reported in at least like 31 states. So that is like their overall distribution looks like. So let's see how this critter looks like and how can we identify a red-headed flea beetle. And as the name suggests, the red cute little head is something that of like, an identifiable character. So they have a reddish head and they are really tiny. I know this picture is a little bit like the bigger picture, but in reality, they are like very tiny pests. So their adults are 3.0 to 6.25 millimeter long. And you can see this is like, they have like an enlarged femur, and this mostly all other flea beetles have, because that is how they kind of like, hop on or jump as you touch. So this is the femur is the third segment of an insect leg. So that is a little bit like enlarged. And as I said, that will help them like, hop and move. And if you see the antennae, it's serrated or tooth-like antennae, and that is also like light brown color. So that is also identifiable characteristics of them. And they have overall like a shiny black exoskeleton and overall like metallic black body. It's an oval shaped, and as I said, adults are like between like 3 to 6 millimeter long. And when we talk about the immature life stages, so this is how the larvae looks like. And it's like a creamy white color, and they have like a brown head capsule. And we have like 3 pairs of legs right here. And then they have like a upward projection towards the end of the abdomen. So that is also a identifiable characteristics of the larvae, and that will also have like fine hairs. So there are 3 larval instars, and the eggs are like again creamy white and oblong. So the larvae, they do feed on roots, but in ornamentals there's not much evidence of them like having damage on roots. But studies have been observed in cranberries that the larval damage is also causing problem in roots. But for so far now, based on the data, adults are the most damaging life stages for this pest. All right, so let's see like how their biology looks like. And so the adults, like they lay eggs inside the growing media of the potted plant, and it's laid inside, and the eggs are the overwintering stage here. And that probably can see between December to March. So I do not have data from Tennessee to speak about, like, authentically about the phenology that we see in Tennessee. And this is based on data from Georgia and partially from North Carolina. So, wish we can like relatively closer. But yeah, in the future, when we do phenology studies in Tennessee, it might little bit change, but this is like an overall phenology data from other southeastern United States. So December to March is when they kind of like overwinter. And as I said, like eggs are the overwintering stage. And after like, when it's like April, all the way till July is when the larval stages are active. And as I said, like we have like three larval instars here, and all these stages are in the soil substrate. So it's under this potted like in the soil substrate in the potted plant. So then there's a pupal stage, and the adults emerge like when it's like the first activity is seen sometimes in May, and it is active all throughout October. So this adults emerge and then they start feeding on the leaves. So this is their overall biology looks like, and Karla will be, talking more about like the degree day models and how to calculate the degree day models. Based on that, we can have like an idea exactly or nearly approximately like what at the time we'll be able to see these stages in for management perspective. So this is their overall biology looks like. So when we go to damage, you can see the first picture here. It starts like, they scrape off the upper and the lower surface of the leaves, and the damage like starts like this, tiny. And as they kind of like feed extensively, you can see that like, it expands. And these tiny black frass-like is probably something that you will also see with the red-headed flea beetle. So yeah, and then as the damage progresses, the short hole is a typical of red-headed flea beetle damage. Like you can see like leaves with so many short holes on it. And then the later part is the skeletonization. So only the midrib will be left, and rest all of the foliage will be eaten. So yeah, as I said, the adults, they feed on the epidermal cells. So they kind of like scrap it from top and from above. And as the feeding continues, it turns out to be like skeletonization. So everything is eaten up. So host information, as I said, like they are highly polyphagous, which means that they have a wide range of like hosts, and weeds serve as an important reservoir host. So, and also the primary impact, as I said before, is the foliar damage, like short holes and skeletonization, similar to, the skeletonization similar to Japanese beetles damage. So, but this is like, you can also probably see the frass associated with the damage. And these are some of the major host plants that they have, like, they really like to go hunt. And growers have observed, like, hydrangeas, roses, like Vigila, then Virginia sweet spires. Like these are, even though they have like they can feed up to like 50 plant species, like including like ornamentals, vegetable fruits. Like these are some of the major hosts that they really like to feed on, even though some studies have found they don't technically prefer, they don't have like much of a preference for hydrangeas, roses, and Virginia sweetspire, but still like growers notice like they do really like these host ranges, host over the others. So, and there's also within like hydrangea, like the panicle hydrangea gets the damage like at least 5 to 6 times more than the serrate hydrangea. So within that is also like noticeable difference between the so it could be like, we don't know like the exact factor why they are really liking it, because it could be due to their the waxiness in the leaves, it could be due to the toughness, or it could also be the practices. Sometimes, like, if a stalk is like infested stalk is kept near the uninfested one, so it can also like transport that way. So a lot of maybes, but yeah, a lot of studies needs to be conducted, like why it has a preference of host over some. So these are major host plants that they love to feed on. So where are these populations coming from? So as I said, like, they have like insect pests like this that has like a wide host range. It could be from anywhere, right? Like the woodlots, or it is like maybe like the near other fields like agronomic crops or pastures, or maybe from we want to narrow it down to mainly like maybe it could be the container plants or the preferred host or the high plant density that is there. So this was a very interesting study that is published in the Journal of Economic Entomology, where they wanted to know if the growing medium is the one thing that has this population like bloom. Is it all coming from the growing population, or it's also like coming from the outside host? So this was a study again conducted in North Carolina, Georgia, and Virginia, and this was conducted in hydrangeas. And in one, they kind of like caged the whole canopy, and the next one they caged the whole plant, and in the other one they just like left like an uncaged plant. So they were trying to see if the damage is mostly coming from the adults emerging from the container. And they also found that, like, yeah, the one that was caged, the canopy was caged, has less beetle damage compared to the ones that is the caged, which means that it has the avenue for the beetles to rise from here and start feeding on it. So this was a very interesting study, and the key observation they found was that growing media is a major source of damaging adult populations in ornamental nursery. So that is actually a great way to begin with. Like, even though the larvae are not very damaging stages, this can be a management strategy, because, and I hope Karla will be touching base on different management strategies. So yeah, but I mean, practically scouting for larvae is a bit challenging. But yeah, this ways, if it is like, we can take care of the larvae, that is actually a great way to tackle this pest. Moving on to the economic impact, so there are several foliar feeding that renders, like, container plants unsaleable. So it's, even though it's the aesthetic damage, people do not want to buy a rose or a hydrangea that has like so much like redheaded flea beetle damage. So, and the heavy feeding, sometimes it needs like, the stalk being laid back, or it needs pruning or regrowth period. So those are also like labor intensive processes that needs like additional insecticide sprays and stuff. So extended crop holding also increases labor and opportunity cost. So this was again a published study where they calculated like the management cost approximately 2,637 per hectare per year from damage from flea beetles in nurseries. And we need to have like multiple insecticide applications. So overall, the economic impact from this pest is becoming a major concern, like over the years, and that is something that we need to be like paying attention to. So this is all I got from my end, and we have like a fact sheet that is solely on red-headed flea beetle, that is a combined product of UT and TSU, and Karla, her postdoc, has also contributed to this fact sheet. So yeah, that is something you can read and figure out, like, if you want to know more about this pest, and if there is any questions, and yeah, we'll have some time towards the end for questions. But yeah, this is my contact information and my lab website, and yeah, I think we'll roll over to Karla now. Stop share.
You, Midhula. Just so everyone knows, I did also put the link to the PDF Tennessee Extension fact sheet in the chat. So you can download it directly from there. Okay. I'm going to talk about management options for redheaded flea beetle. As Midhula said, this is mainly a container issue, and so the management practices are going to be focused on containers. This insect it's native to North America. It's been here for a long time, and it's a little curious as to why it's only started to become a problem in nursery more recently. There's some questions to answer about that and why it seems to prefer laying eggs in containers over soil. But so if you grow mostly in field production, your issues, you may not see this problem at all. And if you're growing containers, you may not have seen this problem, but we want you to be aware of it because once it gets into a container production, it seems to kind of flourish there. So I'm going to cover some items about scouting, about timing treatments, some treatment options, and I also want to talk about gaps in knowledge. There are questions you might have that we just don't have the answers to right now, and we're going to, Midhula and I and some of my colleagues are going to try to do some research on this to answer those questions. So as was already mentioned, there are a lot of hosts for this plant, this particular pest, but our first recommendation for preventing an infestation, if you don't have flea beetles yet, to keep an eye on the plant material that you are bringing in, particularly if that material has soil. So if you're talking about plugs or smaller plants that you're going to be potting up, if you've got bringing in some of the major hosts from other areas like Itea and hydrangea especially, make sure you're looking at the soil that you're bringing in. After that, also looking at if you've received plant material before and you had an infestation, if you know what source you got that material from, if you're getting more material from that same source, maybe make sure that any material that you're bringing in from that location, you take a look at. And if you know that you're bringing in material from certain regions, so we're getting some more hot spots kind of in Virginia, North Carolina, Georgia, that are having their larger container production areas. They have more problems with red-headed flea beetle. And so if you're bringing in material from those areas, you may want to just double check those shipments to see if you're seeing larvae in those material. So since I asked you to look for stuff, I'm going to tell you what you're actually looking for. Midhula showed some pictures of the larvae, and I've got a picture of a pupa there. What you're going to do is look for this larva. I think she already mentioned there's kind of a little finger, little tail at the end of the larva, and it has a brownish, hardened head capsule. And this is important because a lot of people, the first thing you're going to see if you do have issues in containers, you might see fungus gnats. And so I want to make sure you're not confusing them with fungus gnats. So that's what fungus gnats look like. The larvae are like little worms. They're not going to have legs. Midhula already pointed out that the red-headed flea beetle larvae has like 3 sets of legs. These fungus gnats are flies, so they're maggots. They don't have legs, and they have this little black head, and they're kind of white and creamy looking. So just to make sure that if you do see the ones with the little black heads, those are fungus gnats. You may want to manage them as well, but in this context, we're just talking about flea beetles. And so we don't want you treating for flea beetles if you don't have them. A couple other things about container arrangement. So if you've had problems before with flea beetles, you might want to consider separating your host plant blocks with non-host plants. So things that they don't seem to like are the evergreens, boxwoods, things like that. So if you have an option to break up your the host plants you see a lot being attacked with non-host plants, that's a good idea. Also keeping host plants away from weedy borders. I'm going to make some more comments about weeds later, but that might be a good idea if the concern is that they may be coming from the edge of the farm. And also, if you do have a problem in a localized area of the container yard, avoiding putting other susceptible plants that you're newly potting up next to those already infested blocks. So if you have the choice, put them further away so that these beetles do fly, they do disperse. There's not a lot of information about how far they will fly, but they can get through the entire field. So the more, if you're managing an area and you want to move those plants to another area, that's a good way to at least reduce the chances that they're going to start a second population in a different part of the farm. So just some thoughts about container scouting. As I mentioned, you may not be able to look at everything, but you can pick a couple of the things that are the most high value that you've seen problems on before. If I was going to give a suggestion, it would be Itea and Hydrangea paniculata. The Itea gets hammered, so if you do grow Itea, if it's there, it's usually on that first. I have seen it on roses in the Walmart here in McMinnville, so it will feed on other things. I've seen it on some dogwoods. So the fact that it will feed on some things doesn't mean it's going to be preferentially there. So one of the questions we really don't know is how well the Japanese beetle or IFA container treatments that are required for the harmonization and fire ant management. We don't really know how well those treatments work or how long they last for red-headed flea beetle. There is some evidence that those products are probably preventing red-headed flea beetle infestations. So if you grow containers and all of your plants are treated for JB and IFA, and you've never had a problem, that might actually be why. So if you have some plants, especially native plants, a lot of people who grow Itea don't do the soil treatments because they're growing for native or non-neonic treatments. You may want to focus on those types of containers that are not pre-treated. Again, areas of the farm with previous infestations. If you had plants that were damaged last year and you were trying to manage the population, you want to check those plants early in the spring to see if there are larvae present there so you can get ahead of the next year's infestation. Just a note that adult feeding is patchy. It's not going to be uniform across the whole farm, especially with the diversity of plants that we have. So you may have to move around to different areas of the farm to sample and actually see if you've got a problem. We have some advice from North Carolina on how and when to scout. You want to scout containers on a warm day, about 30 minutes to an hour after irrigation. So you don't want it to be cold out, and you don't want the root ball to be dry because the larvae will migrate to the interior and you're not going to see anything. With a nice moist root ball, you can pull the root ball out of the pot and look at the surface on the outside and see if you observe any larvae. They also recommend that you just stand in the full sun because these guys, the larvae, like to be in full dark. So as soon as they are exposed to sunlight, they like to migrate into the root ball again. So it's a little bit of a dance there to make sure you're seeing them and they're not just running for hiding place back in the pot. And their recommendation in North Carolina is to scout 25 to 50 plants per species or cultivar. It's unclear to me exactly how large their plots of they're discussing here, but again, if across your farm you want to, don't just look at one plant per plot of containers. You want to look at several to see like overall if there's anything going on. In addition, so this is that's for early season scouting for before the first generation of adults comes out. Once adults are present, you can do foliar observations. We've observed that the adults like to hide in the sun. They don't like to be in full sun when it's really hot midday. They like to kind of hide in the whorls of the new foliage. And so you could take a look to see if you see any. But they will jump away. These are called flea beetles because they are jumpers. So if you disturb them, they jump, and then you might have seen the jump, but you can't find the adult again. So you're looking for that shot hole or the feeding damage on the leaves. You can also sweep the container yard with a sweep net. This is something we do for research purposes. I don't know how many people would be interested in doing that, but I'll talk about it just in case. These are just some crepe myrtles with damage. And, in this case, this was the most tasty plant in the area. So we had maples they didn't really bother with. We had a few dogwoods, and then crepe myrtles they were more interested in. On the left is an Itea that got hammered. It was sitting literally right next to, on the middle photo, of a Fothergilla, which they seem to not really be interested in very much at all. There's a little bit of feeding damage, like a beetle was saying, hey, let me taste this and see if it's worth anything. And they were like, now we're going to hang out on the Itea. And just that close-up photo that I think Midhula also showed is that they're kind of like rasping the top surface of the leaves, and then the tissue's dying, and that's what ends up causing the holes. And then as the leaf tissue is growing, it's becoming deformed. And the heavy infestations, like you see on the left, can cause stunting. So like at some point with the adults, you have to manage them or your plants are not going to grow properly. So sweep netting is often used in different crop systems in order to estimate, whether or not you want to treat. There really is no action threshold for ornamentals. In cranberries, what they say is if you catch 15 red-headed flea beetles with 20 sweeps, so you're walking through a field at a normal pace and you're sweeping the net back and forth kind of across the top of the plants. If you catch 15 red-headed flea beetles in 20 sweeps, you want to treat. I caught maybe 75 in 10 sweeps at this location one time. And so that's why those plants look so terrible, because the population builds up, and then the adults will defoliate some of the plants. So I want to talk about timing of insecticide treatments, and I want to start by explaining growing degree days, in part because we have not had a lot of issues with this pest before here in Tennessee. So a lot of the recommendations you might read are coming from Michigan. They're coming from, further north, as this insect has kind of moved south in its range. And so if they give you a month where they say, oh, this happened in June, just intuitively you should know, like, we're warmer, so it's going to be earlier in the season, but how much earlier? So for insects and also for plant development, we use a system called growing degree days. And the things you just need to know about it is insects need heat to develop. When the average daily temperature is below 50 degrees Fahrenheit, most insects become dormant. They stop developing. And this is sort of that winter. It's not hibernation. It's called diapause in insects, but it's similar, where they just sit and they don't do anything. But when the average daily temperature gets above 50 degrees Fahrenheit, insects will start developing. So the eggs will start hatching. The larvae will start feeding. And so we can use that information to kind of estimate when we'll see insects, especially these type of insects that are overwintering as eggs. We can estimate, well, how much heat do the eggs need before they hatch? And how much heat do they need before they become adults? And that's what this growing degree days is going to tell us. And the amount and speed of development is related to heat. So like middle of summer, insects develop much faster because it's a lot warmer. In the spring, they develop much slower because it's cooler. More heat equals faster development. So I'm going to explain briefly how we calculate this, and then I'm going to tell you why you don't have to do any math and you can let an app do it for you, because the less math we have to do, the better. So calculating growing degree days for insects, usually we start on January 1st of each year, and you get the average daily temperature for every day. And any day where it's above 50 degrees, you add up the extra degrees. So just as an example, the first 10 days, this is McMinnville temperature, the first 10 days of January, the first 5 days was under 50 degrees, so we had no growing degree days. And then the 6th, 7th, 8th, 9th, and 10th, we had a little bit average over 50. So if we wanted to know what the growing degree days was on January 10th, we would add 0, 0, 0, 0, 0, and then plus 2.7, plus 3.3, plus 8.6, or 6.6, 6.3. I actually added things wrong because I missed the 6.3. But it's going to be around 17 to 25 growing degree days. So make sure you do your addition right. But the good news for us is that we don't have to do the math. There's a lot of different apps that you can use on your phone. What I like to use, just because it's easy, is Syngenta's Greencast. It's a website that I will give you the link to. And all you have to do is go in and enter your zip code. So whichever zip code your farm or plot is in, it's going to ask you the base temperature, and that's 50 degrees Fahrenheit for our insects. And then it's going to ask you the date range. So I did this on Friday, and I put in January 1st of this year to the 13th. And it's going to do the calculations for me. So again, those are the 3 places you need to enter your data that you want. And then it's going to pop out the year-to-date growing degree days, which according to this website is 36. Now, we don't know what weather station this app is grabbing. It's taking presumably NOAA or, Weather Underground, some weather database, and grabbing a weather station and calculating using that weather. But it's going to be close enough for you to make some decisions on. And I use this app, and I actually went through. Okay, there's the QR code for the website if you want. But if you just want to Google it, it's Syngenta Growing Degree Day Calculator. And this is not a promotion of Syngenta. It's just that's the one that I've used before. There are other ones. So I use that same app and I threw in different zip codes in the area of Middle Tennessee. And, as you might expect, Franklin County areas were a little warmer. And then there's a little bit of variation across sort of the production region here in Middle Tennessee. So there was a variation of about 9 growing degree days from that calculator. So it is grabbing different weather stations. It's trying to find the weather station that's closest to that zip code. So you can go in there and, take a look at where you're at this week. And that could be helpful if you have any of the resources that give you pest or calendar date or growing degree day dates for other management, disease management, pest management issues. You can kind of keep track on where things are. It doesn't change a lot every year, but anybody who has lived in Middle Tennessee knows, like, sometimes we have cool springs, sometimes we have hot springs, and it can move about 2 weeks, depending on how much heat or how cool we have. So it is useful in that it'll help you to not miss something or miss a window when you should be doing some type of treatment. So when you're thinking about treatment timings, if you have a previous infestation in containers, you need to treat those containers. Knocking out that overwintering generation of eggs and larvae is going to save a lot of painful cleanup of adults later. The larvae are easier to kill than the adults. And so you can kind of break the cycle of these outbreaks by taking care of the overwintering generation. Now, the data we have coming out of North Carolina and Virginia is that the overwintering eggs hatch around 250 growing degree days. And I'll tell you more about that in a second. They say that that's sort of in shade houses and in cover. But if your plants are overwintered outdoors, it might be closer to 400 growing degree days. So somewhere in that window is when the eggs are going to start hatching. Now, the good thing about this calendar that I like, this app, is that you can go back in time and look at last year's growing degree days. So that's what I did to try to figure out, like, well, when was 250 growing degree days last year? So I took the 2025 year information, and I took it all the way to April. And it looks like around April 1st is when 250 growing degree days was in McMinnville, or at least in 37110 area. And around April 18th was when 400 growing degree days was. So somewhere in those first 2 to 3-ish weeks of April is when we should expect eggs to start hatching. So that's when you want to treat for the larvae. My lab is verifying some of this information by putting data loggers out outside of the shade houses, in the shade houses, and actually in the pots of the plant material, just to see, like, are those numbers coming out of North Carolina accurate for us? They probably are, but we just want to verify so that we have a better idea of what is happening here in Tennessee. So now for some treatment recommendations. As I mentioned, the larval treatments are going to be really important, especially if you've had them a problem before. And if you have cuttings or plugs that come in and you see some larvae, you're probably going to want to treat them before you pot them up, or as you're potting them up. And so that's going to be, again, somewhere between 250, 500 growing degree days. And for us of last year, that would be April 1st through the 23rd-ish. You want to kind of be in that zone. Couple thoughts. Once the first generation of adults are present, there's just going to be overlapping generations of larvae, because they're just going to start laying eggs. And then every 2 to 3 weeks, depending on the heat, you're going to be getting more adults coming out of those containers, which is why I'm telling you, like, stop it before it happens. Because once the adults just start cycling, you're going to have a continuous problem over the summer. And by the end of summer, you're going to have like a huge population buildup. So some of the products that they're recommending for larval treatment, again, incorporations of products. The ones highlighted in yellow, if you're trying to do treatments that are either not neonics, or you're trying to do treatments that are organic. If you're trying to sell to, for certain native plants, you're trying to sell products where you're not using harsher chemicals. There are organic formulations, I believe, of azadirachtin, of the fungal pathogens and the nematodes. I'm not sure if Steiner nematodes, if there's an organic formulation, I'll have to double check. But the thing about the organic options is that you usually have to apply them twice. And so whereas the incorporations of acephate or imidacloprid or the bifenthrin incorporation, you may be able to just do once. Now, I've gotten some conflicting information about like a bifenthrin incorporation, where it looks like they're recommending it for adults, but it's not, nobody's tested it, it seems, for larval treatments. I don't know why it wouldn't work, but that's kind of one of those questions that we still have up in the air of whether bifenthrin incorporations are effective against the larvae. So screenshot that or take a photo of it before I move on. Adult treatments, there's more options for sprays. Again, after 400 growing degree days, and then just like for the rest of the year, there's going to be adults around. So you want to try to knock these out. There are a lot of options. I've had conflicting reports about whether a lot of these things work. The issue what I've seen with the flea beetles, especially for things that they need to be have contact with, is that midday, like, they're hiding, especially once it's, heavy summer. They don't seem to like the super duper heat, which makes some sense that since these are all the way up in like Michigan, they kind of came south from up north. They're more northern adapted insect. And so it may be that, early morning or later in the day when they're happier to be out and on the surface of the foliage is a better time to spray for them. But that's something, again, that kind of needs more research, especially in the South, since we don't really have much information about them. And then again, Beauveria bassiana is the one microbial product that was recommended, and would sort of fit that niche of like if you are trying to avoid conventional chemistries. I do want to talk about pesticide resistance management in the context of this insect. So when we reapply the same pesticides over and over and over again, insects become resistant. It's basically like anything else. Or you eating hot sauce? If you eat hot sauce enough, you need to put more and more on it because your tolerance to it becomes higher. So for these insects, unlike fire ants or unlike Japanese beetle, they don't seem to be out in nature reproducing at as high a rate as they are in the containers. And so if they're sticking to the containers and most of the infestation is coming back from the containers, you're going to be selecting for insecticide resistant populations if you're just applying the same things over and over again. So you want to make sure that you're rotating pesticides. And so I'm going to share with you again a insecticide poster with the different insecticide group numbers. If you're not well versed in like the different classes of insecticides and what group numbers they are, the thing you have to remember is you can't just rotate the type of product. You have to make sure that product is not in the same group. So you don't want to rotate neonicotinoids. So imidacloprid and dinotefuran and thiamethoxam are in the same group. So if you just rotate those three, you're not really rotating. It's going to be doing the same selective pressure. You want to do carbaryl and imidacloprid and bifenthrin because those are in different insecticide resistance groups. This is an example of a poster that CPRO put out. I believe they have a new 2026 one. And this one does have all sorts of different pesticides in the different groups. It's not just their proprietary products, which is why I like this poster. But it gives you examples of products and active ingredients. And there's also a column for flea beetles, which gives you some ideas of which products would be effective. I did look at this and it does seem to be for adult treatments, not necessarily the larval ones. But there are some books that also have this information. And of course, reaching out to any of us for more information if you would like us to help you develop a program. There was a recommendation for an example rotation for flea beetle adults, which would be bifenthrin, whether Onyx Pro or Tallstar. This new product combination, sulfoxaflor and spinetoram, I believe Expire is the only labeled product with that combination right now, a cyantraniliprole and tolfenpyrad, so that you're not applying the same active ingredients each time. But there are other products you could sub in here if you wanted. Okay, I do want to just go through some unanswered questions that are things we're going to need to be looking at in the near future that may help us provide some better recommendations. The big thing is, we've been working for a number of years, and obviously everyone who has to ship outside of the fire ant quarantine zone or are part of the Japanese beetle harmonization program. A lot of people are using container incorporations for those treatments. It's unclear how well those treatments work at preventing red-headed flea beetle larvae and if the longevity of those products is the same or similar to Japanese beetle, for instance. We would hope that it would be about the same, but again, because of the different biology of these two insects, it's possible that the flea beetles could become resistant to some of these products more quickly than like a Japanese beetle would. So that's a question we don't currently know. But if your neighbor has some flea beetle problems and they're growing things without these incorporations and you're growing stuff with incorporations and you don't have any flea beetle problems, then my suspicion is that it's working. But we still have to answer that question. One of the things we don't know is whether red-headed flea beetle will lay eggs in containers with non-host plants. So in our container production, we've got a lot of interspersed plant material. We have been at farms where it's like, okay, here's some hydrangeas, here's some boxwoods, here's some other thing. And the flea beetles are just hanging out in the area of the plants that they're feeding on. What we don't know is whether they'll go in and lay eggs in the neighboring pots, or if they're just feeding and then going straight down, laying eggs, and then coming straight back up. That's a behavioral characteristic of an insect. And so we need to know that, in part because, I'm going to tell you to treat all of your containers that were infested, but I don't know if we need to treat adjacent plants, or if there's a spacing issue, or if we space them a little further apart, then we would be safe. So we don't know that quite yet. Another thing is there are weeds that these beetles will feed on in the wild. We don't really know if they will feed on containers that have weeds in them. So maybe like they won't feed on a boxwood, but if there's bitter cress in the boxwood pot, they'll feed on the bitter cress and then lay eggs in there because they're feeding on the bitter cress. So we need to look into that to see if that's an issue that could be both managed with weed management, but also that it would be even more important to make sure that your susceptible plants are free of weeds, and the plants adjacent to those susceptible plants. So interestingly, the red-headed flea beetle seems to really like pine bark. It doesn't, Japanese beetle doesn't seem to really like pine bark. So different beetles and different larvae have different preferences for the type of substrate they want to be in. So it's possible that different types of substrates or different soil amendments may make the container just not as acceptable. That's something we could look into to see if that is a way to reduce the number of larvae that are in certain susceptible cultivars. We also don't really know if they do better in sort of like light colored pots or dark colored pots, or if the air pruning pots may just dry out faster. They seem to like really wet areas. Up north, they're kind of considered like a problem in more marshy, like wet soils, which is odd, but that just might be the case. And so considering that the recommendation for actually counting the larvae in the containers is to irrigate them heavily first, because otherwise they just stick in, inside to the wetter part of the root ball. There might be some aspects of container choice that may impact red-headed flea beetles. And then looking at surface mulches and things like that people are using, like will the adult even climb down in through the rice hulls or something like that, or will that just be something that they will avoid? And by the same token, if there's larvae or adults in the container, will they be successful? If there's different types of mulch, will they be able to emerge from the pot? And then we've had sort of back and forth with people about whether the flea beetles are actually maybe overwintering in the field border in weeds or soil. Our colleagues in Georgia, I think Midhula mentioned, they did some research and found that everything is just coming back from the pots. So they're going into the pots, they're coming back out of the pots. It didn't seem like there was anything, any edge effects or things like that. But we have different soil types here, we have different weeds. And so again, sort of looking at, could you have a reservoir of some of these insects? Like, even if you cleaned up your entire container yard and everything is gone and everything's clean, like, are there some in the border areas? And so some of our common weeds, we probably want to take a look at and see if they'll feed on them and reproduce on them. And there's an ongoing question about host preferences and whether there are some things that are just going to be more susceptible. We do think like Itea and some of the paniculatas are more susceptible, and maybe that can actually help us in that we don't have to scout everything. For adults, if you're just monitoring the situation, want to keep an eye and see if you have any adults, maybe you only have to look at the Itea you're growing, or maybe you only have to look at the paniculata, because if they're on there, they're going to be on, if that's preferred, they're going to be on other things. So we kind of want to have a better understanding of what we want to keep our eyes on the most. And finally, again, we've kind of had the indication that the root feeding by these larvae are not as problematic, and they're not really stunting the plants that much. But I don't really know if that's true or if it's about, how big of an infestation you really need before the roots start being impacted. So I do think that is important to know, though it's much more obvious that the above ground foliar feeding is going to cause, stunting damage. But still, the combination of the above ground and below ground could end up causing either more damage or make your plants more susceptible to root rots or other types of root diseases. Okay, so in summary, scout your material that's coming in. So you make sure that at least if you're clean now, you aren't going to be responsible for bringing things in. Things could still show up, but at least you're checking your material. Scout those susceptible plants and previously infested material when eggs begin to hatch and treat them. And you want to get that overwintering generation and kind of break the cycle before you start having buildup. And your standard pesticide incorporations in container media that are being used for harmonization and IFA quarantine may be protecting or have some protected level against larvae. And so you may already be doing something and not even realize you're protecting yourself from this. And all of those sort of open-ended questions, we need to do some more work to answer them for you so that we can give better recommendations on what you need to treat and what kind of things you need to do in order to better prevent these types of outbreaks. So if anybody needs to get a hold of me, that is my email and phone number. And I think we can take some questions now on the topic if anybody has additional ones. Let me just back out of here.
Thank you so much, Karla. That was a great job.
Thanks.
So does anyone have any questions for Midhula? We'll start with hers. And I believe you should be able to unmute yourself, or if you're having trouble talking or you don't want to, you can also write them in the chat. So I'm not seeing any for Midhula. Do we have any for Karla? I have a question, Karla. So when you were talking about pest resistance, development from repeated applications of some of the typical pesticides, is it true that with biologicals, like with the Bavaria bassiana that you mentioned, which is a fungus, isn't it less likely to develop resistance to other organisms rather than a specific chemical?
Yeah, it's a little more complicated when the insect is dealing with a disease. It still is possible that they could develop some resistance, but it sort of takes a little bit longer, I think they found. And I'm off the top of my head, I'm not really aware of any insect that has developed resistance to a microbial product. However, they're not meant to be a cleanup product. They're meant to be like a preventative. So if you're considering using those as a drench in containers for the larvae, you want to apply them early when the larvae are smallest and newly hatched, because they're going to be the most susceptible, and there's going to be time for the infection to spread in the container. And so they're usually, however you want to use them, whether it's in greenhouses, foliar sprays, or other things, the earlier you use that product, the better. Yeah, before the population is truly problematic.
But you want to get those spores into the substrate.
Yeah, and into the population, because a lot of the microbes, when the insect dies, it repopulates the microbes into the soil or container substrate. So not with every single insect, but like you'll get more spores. And so any insect that comes into contact with the other insect is going to get infected. So it just helps get that spreading. Just like all of us getting the flu this winter, because we all went and used the same shopping cart. When they're rubbing up against each other, they're passing that disease around to each other.
Well, thank you. And to kind of go along with that, some of the biological products, do you know if any researchers have tried using like some of the essential oils, like peppermint or garlic or anything like that?
No, I mean, as a drench product, it would probably burn the roots, so like that type of application, I probably wouldn't recommend. I haven't seen any products for foliar sprays either. And my initial thought is like a lot of those products for beetles at least are kind of repellents, and tend not to be because they could fly away. So like if they dislike it, they might move away, but it's because of the volatile odors. And so as soon as the smell dissipates, they might come back. So it might just be a very temporary treatment. I don't know if Midhula is aware of any like botanical type product.
Not for flea beetle, no.
Yeah.
Yeah.
We did have one question in the chat. Are you aware of research that has shown there's a relationship between the leaf surface, I guess, type with attacks, like, or like leaves that are pubescent or waxy, being less, having less damage?
No, not specifically, but I think that's an important thing to look at. I think that would be it. And also the thickness of the epidermis, since that's kind of what they like to feed on. Looking at like, they like the itea, they like the paniculata, and those tend to not be as, have as many trichomes. They do, but, so that could be part of, if we find that they just won't feed on oak leaf hydrangeas because they just don't want a mouthful of trichomes. That could be part of what makes a specific cultivar more or less desirable. But it seems like at least in the cage studies that have been done previously, like if you hold the beetles on a plant, like they'll try a lot of plants. But in an open system, they'll move around a bit. But yeah, I'm sure wax and the amount of trichomes has some effect on their interest in feeding on things.
Right. And maybe there are other like chemical compounds in the leaf, like the volatiles, how they taste.
Yeah.
Which I know is right up your alley.
Yeah, there's a lot of plants though. But yeah, we're gonna look at some itea and some hydrangea. And part of it is finding a plant morphologist who wants to do like microtome slices of leaf tissue and actually do comparisons of the leaf tissue for this purpose. So we'll see. We might be able to convince somebody to do that work with us to try to explain. Like we can tell people that they like this or they don't like this, but explaining why they have those preferences is a little more challenging.
Okay. Did anyone have any other questions? Checking the chat. Lee Rumble said thanks. We got a few thank yous. So again, I think Karla put the link in the chat to the fact sheet that she and Midhula had written, as well as Kripa, or Karla's postdoc. And so it has a lot more information there. And if you have any other questions, feel free to contact myself or Karla or Midhula. And I just one plug, we are having our field day here at the Nursery Research Center with Tennessee Nursery and Landscape Association on May 7th. So please put that in your calendar. Midhula, did you have any events you wanted to plug? while we have people
Not,
on?
not me.
Okay. Okay, what about you, Karla?
I think that's the most important one coming up for us, so.
Yeah, we'd love to have everybody there. So, and we follow us on Facebook. And we look forward to seeing you at other virtual events or in-person events. Thanks for tuning in.
Thank you all, bye.
Thank you.
Thank you, Midhula.